Hi, I'm Natalie from Sugar Research Australia, and today I'm very excited about the webinar with Professor Robert Henry. Robert is passionate about his science and about sugarcane, and I can't wait for him to share his latest research. So let's go.
Good afternoon, everyone. I am Natalie Piperdis, and it is my privilege to welcome you to the SRA webinar series. I'm delighted to see so many of you again that joining us today. And I'm confident that today's session will offer you invaluable insight into the latest advancement in our field, and that our must-attend event will offer essential and perspective on the ever-evolving landscape of sugarcane research. I would like to begin by acknowledging the Aboriginal and Torres Strait Islander peoples of this nation. We acknowledge the traditional custodian of the lands of which SRA is located and where we conduct our business. We pay our respects to ancestors and elders, past and present. So this work is a partnership investment between the ARC Research Hub for Engineering Plants to Replace Fossil Carbon, Sugar Research Australia, and the Queensland Alliance for Agriculture and Food Innovation at the University of Queensland. So today's webinar, hopefully will go better than it started, will consist of a presentation for around 45 minutes or 40 minutes and followed by some Q&A session for 20 minutes. So during the presentation, I'll ask you to all mute your microphone, and then we can ask questions during the talk, but in the box. And during the Q&A session at the end of the presentation, we'll get to the question. So without further delay, allow me to introduce Professor Robert Henry. I was really delighted when I asked Robert to speak at the SRA webinar because his enthusiasm for science and for sugarcane and is truly a blessing, and he accepted very quickly. So I'm confident that Robert's infectious passion for science will captivate you, and I trust that you will find his presentation, Engineering Sugarcane to Replace Fossil Carbon, engaging and insightful. Robert is a professor of innovation in agriculture at the Centre for Crop Science at QAAFI. His expertise lies in the molecular analysis of agricultural crops, with a particular focus on Australia flora and economically and socially significant plants. Robert has been at the forefront of research in genome sequencing, aiming to capture novel genetic resources for the announcement of food crops and the development of improved food products. In fact, I suspected that Robert has a few clones and he's everywhere. Indeed, congratulations are in order, Robert, on the recent publication in the journal Nature, which revealed the complete sequence of the sugarcane cultivar R570. This achievement is truly remarkable and a testament of groundbreaking work and to your dedication. However, today's webinar will focus on the project Engineering Sugarcane to Replace Fossil Carbon and the action needed to develop the required technology, infrastructure, and capacity to meet demand for biofuel in the aviation industry. So without further delay, Robert, the floor is yours.
Thanks very much for the great introduction, Natalie. And today I wanted to take the opportunity to talk to you about the work we're doing, particularly in the Research Hub for Engineering Plants to Replace Fossil Carbon, with a very strong focus on sugarcane. So if we could have the next slide, thanks. So the context of this work is that what we're looking to do is find a renewable replacement for fossil carbon. Instead of oil to produce our biofuels, bioplastics, chemicals, all the things we produce from oil. And the important thing to understand is this is not just about biofuels. Most oil in the world is not used for transport or energy. It's used to make things out of carbon. And this is the role we're proposing for lignocellulosic feedstocks, specifically sugarcane biogas as a key opportunity. So what we're about is really the opportunity to genetically improve the composition of sugarcane to make it a more suitable resource for this role. So if we can have the next slide, please. So we've probably often heard about the issues associated with the food versus fuel debate. The reality is that we need food. Food security is critical. We also need energy, and we need to conserve biodiversity. We have to balance all those. I wrote a book back in 2010 trying to tease out the issues around how we balance those things appropriately. But it's not an option to say we're not going to have these products from agriculture, because there is actually no alternative. If we can have the next slide, please. So the processes we're talking about is converting biomass like sugarcane bagasse to the things that we currently produce from oil. And in the past, we've thought about producing ethanol and diesel for vehicles, and to some extent that is still with us, but we're seeing electrification of those energy needs. And so we're going to expect that particularly vehicle transport will be driven by solar and wind power into the future. And so we don't really, in the long term, need a liquid fuel for ground transportation. But we do need a carbon-based material for all those other non-energy uses like plastics, fabrics, clothing, building materials that are made from carbon. And this is a role that will persist no matter how far we go with solar and wind and other alternative energy sources. And the other big category is aviation fuel for long distance flight. We don't envisage that electrification is going to satisfy that need. And maybe the next slide will say something about how we're going about this. What we're talking about is a biochemical process where we can convert the plant biomass through pre-treatment, or hopefully avoiding that with the new varieties of sugarcane we breed, to sugars that are an inexpensive source that we can convert into all of the products we currently produce from oil using microbial and chemical conversion techniques. So the next slide. And so an example that we're using as a prime example of the technology that we want to commercialize is converting sugarcane bagasse to jet fuel. This is something we can't do with solar and wind power. We can't electrify planes for these long distances. And here's the molecule one of the molecules that we favour, and that's dimethyl cyclooctane. This is a superior fuel to Jet A. So if we produce this fuel from sugarcane bagasse, the planes can actually fly further. This is not just an alternative. This is a superior alternative that can be made from sugarcane. And it also will have a role in military applications, for example. The next slide. So this is the sort of plan that the airlines have going forward to try and address the need to reach net zero by 2050. We can see that in the short term, flights over short distances, regional flights, can be electrified. In the next 5 to 10 years, we could be flying to Sydney in electric planes. But as time goes by, battery technology will improve, and it is envisaged that short-haul flights will also become possible with electrification. But you'll see that the long-haul flights, even by 2050, will require a liquid fuel. And this is where we'll need sugarcane bagasse or some other source of biomass to produce that fuel, because electrification doesn't seem like an option. The next slide, please. So if we look at where we can produce the biomass to actually fulfill this essential need as part of the decarbonisation strategy in the medium to long term, sugarcane is probably the best example worldwide. The other key example that's often cited are the eucalypts. They represent a source of woody biomass that might also be used in this way. And the reason that these two candidates stand out is because of their high productivity. We need a very large production of biomass per hectare to justify this. We can't use a large land footprint. We need to produce this on a small footprint, we need 100 hectares of dry matter per hectare per annum type production. We can do that with sugarcane. We can do that with eucalypts. We cannot do that with many other crops. And so it is for this reason that sugarcane becomes the world-leading example of the source of this type of fuel. The next slide, please. So what we're working with are the advances in the underlying sciences, the advances in genomics, the advances in gene editing that would allow us to improve sugarcane to make it a really optimized source of biomass for this type of application. The next slide, please. So gene editing is something that is a very powerful technique that allows us to change. And what we want to do here is change the composition of the biomass so that we get much higher yields of the final product, whatever that product might be. And so we've developed very high throughput gene editing. We're doing that in rice as a platform, and this is a model where we can test what would happen if we were to edit particular genes in terms of composition, and the most promising of those we can take forward to apply in sugarcane. The next slide, please. And the other technology that we've developed is high throughput mutagenesis. So the other way we can introduce the changes that we need are to use chemical mutagens. And this has been a classical approach in plant breeding, but it hasn't been possible to do it in high throughput. What we've developed in collaboration with our Carlsberg partners is a very large population of more than 150,000 lines of sorghum with all of the possible mutations that you could have in sorghum. So from this population, within because it's pre-existing, by screening at the DNA level, within a day or two, we can identify an individual that has a mutation or a knockout in any one of the genes that are present in sorghum. So here we have a possibility of looking at all the genetic possibilities in a single library that's pre-prepared. Now, sorghum is a very close relative of sugarcane, so discovering mutants that have the right function in sorghum is something that we're going to be able to translate very readily into sugarcane. But because we're able to do this mutagenesis more efficiently in the diploid sorghum, this provides the platform of science on which we can build the new sugarcane variation. So next slide, please. So we've been advancing the underlying DNA sequencing technology over the last couple of years and applying it to plants as it's been rolled out. And the advances in the technology have been quite dramatic, and they allow us to look more exactly. If we're going to edit the genome, we have to know exactly what it is. We have to know about all the changes we might make if we go in and intervene in a particular way. So we need perfect DNA sequences. So that's why we've focused on improving this technology. So the next slide. And what we've ended up with is proving that we can get a perfect genome. And the first one we reported was a couple of weeks ago, which was the mango genome. Here we could get a perfect genome just from the data from the sequencer. For many plants and organisms, we've been able to get a genome sequence, but we've had to use genetic mapping and all sorts of tools and knowledge to put together the genome. We're now getting to the point where the DNA sequence data alone is proving sufficient to get high quality genomes. And that sort of innovation is what's driving forward our ability in this area. So the next slide, please. And so we've used that to come up with a better rice genome. By resequencing the rice genome, we've just published it now, we've found another 10% of the genes that were not known until we did this sequencing. So what it shows is that the higher accuracy of the new quality genome sequencing tools we've got allow us to find all of the genes, not just many of them. And that means that we're going to be able to be much more deliberate in what we do in genetic manipulation using either mutagenesis or gene editing. The next slide, please. So we've been working very heavily with sorghum because it is a great model and much easier to work with than sugarcane. And sorghum, interestingly, is a genus that's predominantly Australian. We domesticated one species from Africa, but the bulk of the species are found in Queensland and in the Northern Territory. And so we're now working with an international consortium, sequencing all of those to understand all of the genetic diversity that's available in the whole genus as a source of novel variation that might underpin genetics of this whole group of plants. Okay, next slide, please. Of course, sorghum is a diploid, and sugarcane is a highly polyploid, 12-ploid organism that hasn't been amenable to DNA sequencing, but things have moved ahead. And I illustrate that by our work with coffee. We published the coffee genome, the coffee robusta genome, a diploid, in Science back in 2014. We're just now having press the Arabica coffee genome. It's a tetraploid in 2024. So it's taken 10 years to get to the same point in a tetraploid that we could get in a diploid 10 years earlier. So the next slide, of course, tells us that we've been able to do this with sugarcane. And here for the R570 genotype, we can see the very large number of chromosomes compared to sorghum at the bottom, and even the progenitor Sucarrum species above that. The fact, has already been mentioned, and I notice Karen Aitken's online. We've, as a group, locally and internationally, been able to sequence this genome because of those advances in the science, and we've been able to publish it in Nature because it is the last of the 20 major crops in the world to have a genome sequence, and it's certainly the most complex, and that's why it's taken so long and been the last. But it gives us a great platform. And in reflecting on what's got us here, it's been a very long-term effort. We had a meeting 10 years ago in Port Douglas where we negotiated and agreed finally that we would sequence R570. The Brazilians wanted to do their varieties. The Australians, of course, wanted to do Queensland varieties, but we agreed to do R570, and we all signed an agreement known as the Port Douglas Accord internationally in the sugarcane community, where we agreed to work together to do this, and that's what's resulted. Karen Aitken put forward the final push in this with a engagement of the Joint Genome Institute and the US Department of Energy in providing the resources to get this together. But we've been able to combine the sequencing capacity, the transcriptomics efforts locally, Karen's efforts to produce genetic maps of sugarcane, and Angelique Dehonte's efforts in France to understand the karyotype and the actual structure of the chromosomes in the plant. By bringing all that knowledge together in a single variety, we were able to achieve this. And this now is the platform that we really want to use. Next slide, please. So what we've been looking at is how the composition of the bagasse might influence our ability to convert it to fuel. And we find even small differences make a big difference in these processes. So it does suggest there's great potential for breeding sugarcane that will be better for these applications. What we're about is changing the composition such as a higher proportion of the carbon can be converted to the final product. If we're making dimethyl cyclooctane as jet fuel, we can do that now. But what we get is a relatively small proportion of the carbon in the bagasse into that final product. By changing the composition of the bagasse, we'll get a much higher proportion of the carbon into that final product, dramatically reducing its price. If we think in terms of ethanol, at the moment, we can convert maybe 25% of the carbon that's in sugarcane bagasse to ethanol. What we can see is that by some fairly simple genetics, we can probably increase that to 50% or maybe more. That would double the yields of ethanol, halve the cost, if you like. And so for the jet fuel molecules we're working with at the moment, they currently cost 2 or 3 times what the fossil fuel equivalent would be. But if we can change the starting point and come up with sugarcane bagasse where we can get 2 or 3 times the yield, we can actually make a product that will compete directly in price with the fossil-derived product, and that's where we need to be. People may say they'll change to these products for environmental reasons, but we've really got to do it at the same price. If it's cost competitive, then people will be happy to change. While it's more expensive, I think adoption will be difficult. So the next slide, please. So what we've done is working with the Indian Institute of Technology. UQ's got a partnership there where we have joint PhDs working in the chemical engineering department, working on how to convert sugarcane bagasse to sugars and be able to go down this biochemical path to the whole range of end products. And we've made very significant advances again in the last couple of years with quite new approaches to that processing technology. But one of the objectives of the genetic modifications we're looking to make is to eliminate the need for some of these steps and to greatly simplify our ability to do this conversion. So we're not only going to increase the yield of the product, we're going to decrease the cost of making that transformation. The next slide, please. So we've been very much using modern molecular biology, omics technologies to look at all possible levels in the sugarcane plant to try and understand what we need to do. What have we got? What could we change? What should we change? Where should we go? So there's a whole lot of omics going on in that area. The next slide, please. And so what we're focusing on particularly has been the sugarcane transcriptome, how to understand the expression of genes, which genes are expressed and how does that influence the composition of the culm and particularly the composition of the biomass. And we're getting a much better understanding of that and how it varies within the germplasm. But what we're talking about really is making quite radical changes in getting to compositions that are outside what we find in the natural material at present. The next slide, please. So what we've done is put a big effort into coming up with a reference transcriptome. So this is basically a list of all the genes in sugarcane that are expressed so that we can actually compare different individuals by having a reference to the full set of such genes. And there's been quite a lot of work going into this because sugarcane has got an incredibly complex transcriptome with all those copies of all those genes. But we've made great progress in recent years in getting that material together. The next slide, please. One of the recent discoveries is that we've been missing some of the very long genes in all of the studies across all plants, and that we've found that to discover those, we actually need to be quite deliberate in targeting, developing tools for finding those long ones. And in fact, nearly half the genes are missing in a lot of the studies because the long genes have been overlooked, and we're now correcting that with some of these new insights. The next slide, please. So we've looked in different organs. We get very different genes expressed in different parts of the sugarcane plant, and that explains the difference in the composition of the biomass and the residue after sugar extraction. So we can see that it's possible to have very different composition in different parts of the plant. What we need to have that, of course, is those different compositions in the culm. So the next slide. So we have done very deep work in the leaves, and it tends to be that a lot of work in plants is in leaves. So we have a great understanding of the genes expressed in the leaves and how all of that works. But the next slide is that we've really had to focus on the culm. This is the part of the plant where the action is. We need to understand this in great detail. And so we've looked at how the composition changes through development in a large number of varieties at different internodes, trying to understand the genetic control of bagasse composition in that developmental sense across varieties and time. The next slide, please. So in a large experiment we did with SRA over the last few years, we've looked at a number of internodes, and we've looked at gene expression in more than 1,000 different samples representing different parts of the plant at different stages of development. So we've built a huge set of data, not all of which has yet been mined, which provides something that we can continue to mine for new insights into the sugarcane plant and how it grows and develops and how it might grow and develop in different ways if we were to change things. The next slide, please. And so we can actually identify different groups. The different sort of groups we get tend to come probably from different breeding objectives over time in the breeding program. There's been more emphasis in certain times on having high early sugar than in other times, and so we can see that plant breeding works. We always know that it works, and what we see in the metabolic features of the sugarcane are the sort of signs of that selection, and that we have been able to select different types of sugarcanes, and that gives us encouragement that we can go down particular paths. The next slide, please. So one of the key things that we want to manipulate, of course, is the lignin content of the sugarcane. And one of the things we've shown is that even the composition of the subunits in the lignin is important, and that there's variation in sugarcane, and that variation does influence performance. It's just recently been shown that changing the subunit composition of the lignin has a really significant impact on our ability to process that. And so what we're looking for in all of this is ways that we can change composition without altering plant performance. We want the yield not to change, but we want the composition to change in such the way that we've got much greater processing options. So next slide, please. So one of the things with sugarcane being a polyploid, having 12 or so copies of every gene, is that we've got the potential for allele-specific expression. We might express just one of those copies. We might express 12 copies or some other number. And we've had a lot of challenges in understanding this process in such a complex system. So we've devoted quite a lot of effort to getting an insight into how the sugarcane plant works in terms of allele-specific expression. The next slide shows us a result of some work we did a couple of years ago, where we looked at comparing the level of expression of genes with the abundance of the gene in the genome. So you could have from 1 to 12 copies of an allele, and you could measure the expression of those two different alleles in the genome. And we can see here there is a linear relationship for most of the genes between the level of expression on the vertical axis versus the copy number on the horizontal axis. And this was a remarkably linear relationship, showing that these genes are being expressed in an effectively and unbiased way. If there's more copies, you get more expression for most genes. And so 80% of the genes, it turns out in sugarcane, seem to be expressed in a relatively unbiased way. Further than that, you can see just how accurate this very high coverage sequencing was. We sequenced this large genome to great depth, and we sequenced the transcriptome to great depth. And you can see that this is not continuous. You can see that there's separation of 12 groups. You'll notice along that axis, you've got 12 clusters. You've got the 1 copy, 2 copy, 3 copies, up to 12 copies. So we can actually distinguish 4 copies from 5 copies from 6 copies in terms of both the level of expression and the frequency in the genome. So it shows us that the tools allow us to understand copy number and gene expression in sugarcane in a way that we thought probably we weren't going to be able to. The tools really are up to it. You will see that there are some genes along the top of those figures, and these are genes that are expressed regardless of the copy number, which varies along that axis. They're expressed, only one form is expressed, and this is unlike the other genes that are expressed in an unbiased way. These genes are expressed in a completely biased way. Only one allele of the two is expressed at all. And these turn out to be predominantly defense genes, and that's what you'd expect, whether it's in nature or in plant breeding. Plant defense genes will have been under intense selection, and there'll be great preference for expression of just one of those alleles. The next slide, please. So with that insight, we've looked more deeply recently, and with Prathama from SBI in India, we've used long-read transcriptome sequencing to understand the relative contribution to the two progenitors in sugarcane, working out their relative contribution to the transcriptome and to the traits. And what this has done is confirmed some of the things we've understood about the relative contribution of the two progenitors to the key traits of a modern hybrid sugarcane, but also gives us new insights into how we might reconstitute a modern sugarcane hybrid to have a different combination of those traits. And this long-read technology allows us to clearly distinguish which of the progenitor genomes any transcript is coming from. The next slide, please. So in this complex genome, we can attribute to the specific subgenomes. We do see a predominance of genes influencing the sugar content, being the genes coming from the officinarum progenitor chromosomes or genes, and the spontaneum genes are very much associated with the stress responses overwhelmingly. But we get much more detail by being able to look at every transcript. The next slide, please. So to summarise, we've identified a huge number of candidate genes. We've got a genome, we've got a transcriptome, we know about the differential expression of genes and how that's relating to composition. We now want to test them systematically. We want to find those combinations of genes, gene modifications that deliver the sort of changes we want. And that's what the new research hub will focus on, putting together a large team. The next slide, please. And one of the further refinements, we just had accepted a paper, it's in fruit, but it's on spatial transcriptomics, and we hope to apply that in sugarcane. That allows us to look at transcription in individual cells and across a tissue. So what we'll be able to do is look across the column at the different tissues and cells and develop strategies to alter the expression of particular cell types or particular tissues. And this will give us much more power to be able to change composition without having any adverse effects. The next slide, please. So again, to summarise, we've established genome editing and mutagenesis, and we've got high throughput potential and the option of using rice and sorghum models to screen very large numbers of genes so that we can have smaller numbers of candidate genes that we bring into sugarcane. Our intention is to develop and apply gene editing to sugarcane to bring the most promising of these genetic interventions into the sugarcane genome. Next slide, please. So we've put together a team, I think, that includes all of the people we would need to do this, people with the expertise wherever they are that will allow us to perform this process aggressively, test hopefully thousands of genes, really exhaustively test all of the gene options that might be conceivably able to deliver the outcomes that we want. The next slide, please. We've also got we've put together a very large team of partner investigators, and we've got many more joining. The next slide, please. So these are the partners in the initial partners, foundation partners in the hub, but we've already got many others joining, and so we're looking forward to applying all this technology to coming up with sugarcane varieties that really will drive the development of an industry where we can produce high value products from the bagasse. Next slide, I think. Well, that might be the end. Yeah, so just to put this in a broader context, in adapting agriculture to climate change, I've been involved with lots of different initiatives, and our ARC Centre of Excellence for Plant Success is really about conventional approaches to breeding, stress-tolerant plants, and we're always going to need to do that. That's a mainstay of effectively accepting that if the climate changes, we've got to adapt agriculture to it. But there are other things that can be done, and agriculture can actually help to mitigate climate change. And so we have a new zero net emissions CRC for aiming to eliminate emissions in agriculture, make agriculture carbon neutral by 2050. This is the biggest CRC ever funded, and that's attracted around $300 million. But what we're talking about in the hub that I've been introducing today is something that's actually different. It's agriculture, but it's agriculture where we're aiming to reduce emissions in areas outside of agriculture. It's not making agriculture carbon neutral, it's making the whole system carbon neutral by replacing fossil fuel with a renewable source of carbon. And I would argue one of the best renewable sources we have available is sugarcane bagasse. Thank you. That's what I wanted to say today.
Oh,
Natalie.
thank you so much, Robert. So great talk, very inspiring. So now it's time for question. That is a question from Sam Forsese. Some are looking to Pongamia as a solution to biomass / fuel instead of sugarcane and eucalyptus. It's not really a question, but do you have any things to say to that?
Yeah, so I guess I could comment generally on oil-producing plants versus lignocellulosic paths. There's certainly been my analysis that oil-producing plants are not really productive enough. We have looked closely at the potential of producing oil canes. You could produce sugarcane oil in the sugarcane plant, but the yields that you're likely to achieve would mean that you would just need too much land to meet our requirements. The great advantage of sugarcane is that we can produce very large amounts of sugarcane bagasse, such that this will meet, for example, Qantas's needs for jet fuel, probably from the existing bagasse supply. Whereas if you were to use an oil-producing plant, we would need probably to expand to the extent that we really started to compete with agriculture in a serious way with food production. So it is all about the scale of production and the quantity per hectare. You need to think about how many tonnes per hectare of product you can produce. And unless that number is high, you're going to need a lot of land to fulfill this need. The whole idea here is that we don't create any problem for food-based agriculture by producing this from a relatively small area or from products that aren't used for food, like wood chips or sugarcane bagasse.
So Dylan thanks you for your presentation, and he asks you, do you see a role for sugarcane trash in this space as well?
Well, I think we're all aware of the important role that trash plays. So we would be wanting to be careful in suggesting that you remove it from the field. Clearly, it could contribute, but my overall expectation is that we would still probably favour using the bagasse from the mill and making use of the trash in the conventional way. But there's no reason that trash couldn't go through this process. It's a question of the trade-off between the agronomic implications of doing that.
Thank you. So Robert Doerr, then it is likely both a biotechnology-produced sugarcane be in crop carbon partitioning would preclude this new cane variety from human consumption suitability.
Yeah, so my comment on that is that we do expect that most, if not all, of the changes that we're thinking about making would be loss of function. These would be knockouts, and under current Australian rules, these would not be GM. And we're seeing the food legislation being adopted at the moment. So gene editing that involves introducing a cut in the DNA and allowing natural repair is considered not to be GM. It's something you could have done by mutagenesis or conventional breeding. So we believe that these will not be thought of as GM. So the market implications and human consumption will not be questioned, I believe.
Megan McKenzie's just said, you mentioned using bagasse. Bagasse is being used to generate electricity by mills. Are there other sugarcane materials that can be used? Example, trash, but what is availability and techno-economic benefits?
Well, Megan, the key point here is that most mills have traditionally burnt the bagasse very inefficiently to get rid of it. They need relatively small amount of bagasse to meet their own electricity needs. And in fact, what we're seeing is with the developments in solar and wind, the price of electricity is going to decline substantially. The value of bagasse as a source of green energy is going to be lowered, and this will be a substantially higher value use. So I think that the mills will turn to using solar electricity because it'll be cheaper, and they'll sell the bagasse as jet fuel for a much higher price. So it's about that finding the highest value use for your product. But all the suggestions are that already the threat of solar and wind is making bagasse to electricity less attractive.
We got a question from Adam Douglas. What are the yields per tonne of bagasse to ethanol?
I can't give you a definitive figure. I think my earlier suggestion is that we're at that. It's in the 10 to 20% range at the moment. But what we're arguing is that we can double or treble that relatively easily with the changes we're thinking of.
Ian Biggs, thank you, Robert. Do you think sorghum will be able to provide similar fuel source considering its larger growing environment, or would the modification of sugarcane allow it to grow in drier or more challenging environments?
Great questions, and we don't really know definitively the answers to that. That's why we're working with both sorghum and sugarcane. Potentially both of them could contribute to processing through the same facilities. It really depends how good the sugarcane breeders are at breeding sugarcane that can cope with more marginal environments. If not, then sorghum may have more of a role. But I think inevitably both sorghum and sugarcane will have a significant role in this space in different locations, depending on the rainfall and other aspects of the environment. So I see a strong future for both.
Raul Castillo, how much fibre are you looking for energy canes? Sugar will be the less character worked by breeding programs.
Yes, it might be the case that it really does depend on the price of sugar, doesn't it? And the price of bagasse. Depends how much a tonne the bagasse is worth compared to the sugar. If the bagasse is more valuable, then people might be happier to have higher fibre canes. But we're not necessarily talking about high fibre canes, although that could contribute in this space. We're talking about the fibre having a different composition, so that for every tonne of fibre we get much more of the end product. And so it's a separate consideration to some extent than the proportion of sugar to fibre.
Yeah, thanks, Robert. Any other question for Robert? Actually, I've got a question. Robert, you meant, oh, hang on, I'll just do mine afterwards then. There's a question there for you. Do you think bigger cane always be the best option, or there is a specific criteria?
I'm not sure. I know there's a lot of consideration about what benefits there would come from having bigger cane, but it all depends on your rotation and a whole lot of things. It's a complex question, and I don't know the answer. I'm not sure the bigger cane always would be the best option. It's returns per hectare per annum, which it always is, that you've got to look at.
Right. I've got a question, Robert. So you mentioned the Colombians have sequence as well. So is anyone like comparing like the sequence from R570 and CC and the Colombian cultivar that they're nearly sequenced?
I think I've been involved in a lot of comparative analysis of genome sequences, and I think to do a good comparison, you need sequences that are of similar quality. You can't compare one high quality genome with one that's full of errors, because so much of the differences may be due to the quality of the data. So I think we will see people doing comparative analysis, but I think, and we've done it in a number of other species where we've actually deliberately resequenced a number of genotypes using exactly the same technology, so that we can make that comparison. So we're not comparing different methods, we're actually comparing the genotypes. That will happen now in sugarcane, because I think the advances in the technology that got us to genome sequence haven't stopped. And really, if I look back at now, we could probably do so much of it so much more easily. So I think we're going to be able to produce lots of reference genomes for sugarcane over the next few years as the technology advances. So we're going to be able to answer the question you ask about exactly what the differences are at the whole genome level between sugarcane varieties, and it's going to become increasingly inexpensive to do so. To some extent, having one really good genome does help you to get very high quality, if not the same quality, genomes for lots of individuals, and that allow those comparisons to be made. We're comparing apples with apples rather than something else.
Yeah. Yeah. And I was just like thinking about, like genes comparison and, that sort of things, and starting with like some sequence that for genes that we need, or that sort of things, especially like.
So if you're comparing the genes, of course, you do need a comparable annotation. And so sometimes if the genomes are annotated with different tools, they miss some genes or they find others, and it's a function of the method rather than a difference in the genotype. So again, we try and annotate the genomes with exactly the same pipeline if we're going to compare the gene content of different varieties. But we would expect in sugarcane, we're going to find some really extraordinary differences in gene content between varieties, given that there's so much redundancy in the genome and the potential for material to be chromosomes to be missing in the thing to still perform. So I think it's again to be a very interesting area to see just how much difference we've got in the gene complement of varieties.
Thanks, Robert. All right, so Troy, does the increased carbon content in the fiber alter the sugarcane, or the sugar content, able to be extracted from the plant?
Yeah, so we're not increasing the carbon content, we're changing the forms of the carbon. So we're going to have more carbon in cellulose and less in hemicellulose, more carbon in carbohydrates and less carbon in lignin. So it's not more carbon. So there's no net competition in what we're proposing with the sugar. It's different carbon compounds.
Ian Biggs, Robert, as you build this understanding of the sugarcane genome, do you think you will start to unlock the N2 fixation story in sugarcane?
It would be great if we could. I mean, I think we all understand that somehow or other sugarcane's far more nitrogen efficient than it should be. That we don't really understand, and this is part of the attraction of sugarcane, that it is so nitrogen efficient compared to anything else that I think of. Be great to understand that, because maybe we could enhance it even further, whether it's microorganisms or whether it's something intrinsic in the plant, or whether it's a combination. I certainly don't understand why sugarcane is as good as it is, but it'd be great if we had that insight.
Yes. Anyone has a question for Robert? Oh. All right. Oh yeah. Okay. How far from production are new varieties?
Well, I think that a lot depends on how fast we can get them through the SRA pipeline, I suppose. I don't know the exact details, but the reality is that we've got to do these things in the lab and in glasshouse and get them to the field as soon as we can. It is a matter of how good they are and how much you can fast track them. If we start with superior varieties and make very specific, deliberate changes that improve the value of these traits, I could envisage that would allow the fastest possible tracking of these new varieties through to commercial production. But you're probably best to talk to the SRA breeding people about how fast we could go from a initial plant in the glasshouse to a commercial scale.
Yeah, we got a question from Errol. Have you done this type of studies in Sagrum spontaneum to check the carbon content and exploit its genome?
We haven't tried this specifically in the progenitors, no.
So Robert, you're mentioning, so we're doing engineering, and so what are we, where are we up to with OJTR and that sort of things, or are these modifications are going to be accepted by the public, or what's?
Yeah, so what's happened is that the OGTR has decided that these types of knockouts are not GM, so they're not being regulated as GM, and we're working within the institutions to make these decisions on edited plants in terms of when we release them into the environment, but they're not subject to that regulation. There have been food regulations, but food standards people are redrafting the Australian legislation to exempt these from any food regulations as well. And that should all be in place this year. So at the research level, they're not GM. At the public level, they won't be GM, hopefully within the year. And we're seeing the same thing happen in other jurisdictions worldwide. Even in Europe, where they've been very concerned about GM, we've seen real turnarounds in some of the jurisdictions where it's recognised that these products need to be effectively deregulated, and that's what's happening. So there's going to be great attraction, and really, I think in the end, no real barrier to taking these to market. There doesn't seem to be separate to the government regulation or the food industry regulation any great consumer concern, and of course we won't be labelling. It won't be something that's treated as other than conventional. So I think we're not going to have a problem, but who knows what sort of reaction will be out there in the community.
Yeah, that's right. And I was like looking at one of your first slides. So you'll be those planes that will be electrical will be a hybrid or just electric? Because I'm just kind of, even if I have to come from, not too sure. You might have to try first, right?
I think they are producing hybrids, but in the main, these are going to be straight electric planes for short flights. It's my understanding. If you look at both Airbus and Boeing, I think they both have pretty much the same medium to long-term plans in terms of the way they're going to power their planes. Hydrogen is an option that may or may not also play, but it's too heavy for the long distance flights. So we still need sugarcane for the long flights. So hydrogen may or may not become a fuel for planes, but it'll only be medium distance from the current analysis of the opportunity.
Wow. Okay. All right. Well, I think we don't have any further question. So, yes, there is. Okay. Okay. Okay, so it's just well, no, it was just like to thank you, Robert.
Thank you.
So, well, thanks again, Robert, for an excellent presentation. I hope
Thanks for
I'm
the
not out
opportunity.
of. I hope you can still like give me a lot of presentation. That we got lots of meetings coming, and
Yes.
I hope you all enjoy this presentation, and I can we can all agree that it was very informative. Thank you for your attendance, and I hope you enjoyed today's webinar and that you will join us for the next SRA webinar coming soon. And if you want any information, everything is on the events page on the SRA website. So thank you, and thank you so much, Robert.
Thank you.